Sorry about that.
No, sir.
Okay, thank you very much. So what is the precision medicine model that is on the foundation of what I did? So the precision medicine model attempts to integrate all these different layers from participant computed data through behavioral analysis all the way down to genomics, so that it can create an integrated platform through which in autism we will be able to identify subtypes and develop personalized diagnosis and treatment. However, the problem lies on this level here, the behavioral level, because the way diagnosis and research takes place generally in this field is people assume a one-size-fits-all model. So basically, they assume some a priori distribution in the population. So you have all these different samples from patients and subjects, and by assuming a priori that this whole sample size will follow a normal probability distribution, what basically is done is you compute some theoretical mean. Basically, you have a theoretical mean, and then you assume that it spreads normally around that mean. But this grand averaging leads to a loss of gross data, and maybe in this gross data is where important information lies. So what we did here is we adopted a paradigm that is used traditionally in computational neuroscience, where basically you have digital spikes from musical cortical recordings, for example, and then they pass through a kernel, and we get analog continuous functions that we use to characterize brain activity. What we did here, so we can adapt this to a more general class of biorhythms, and with a big emphasis to kinematics, is we got analog continuous streams of biorhythmic activity, and then we convert this into digital spikes, and that allows us to compute interesting properties and develop this unified statistical framework to connect the different layers. So how is this done? Like briefly, is, for example, here you see a speed streamline a time series, and then you take some window in time that where the process is stationary, and you compute these standardized fluctuations, which we call micromovements, and then you can compute empirically estimated histograms and parameterize the space of all these different biorhythmic activity in order to locate different subjects and different subtypes of disease to some parameter space that explains these empirical histograms. So what is exactly the micro movement spikes, this whole spike process that we're talking about? So basically, you compute the empirical mean from raw data series, and you subtract the empirical mean, which may be different for different subjects and different individuals. You subtract this empirical mean, and then you take the absolute deviations of the process from that mean, and then you normalize the peaks of this activity with the local average, and that's how you get sort of a peak activity and a discrete analog of this continuous process. And we accomplish two things with this transformation. One, we remove any bias that is introduced by anatomical differences between subjects where usual normalization techniques fail, because this process is compatible with the laws of anthropometry. And two, you can do this characterization with different famous probability distributions. And in our case here, and before we move on to the nature of these histograms, let's talk about how we can apply this new framework. So by using this statistical framework that in its foundation is this micromovement transformation, we can characterize an entire functional span of actions in the nervous system stemming from autonomy all the way to involuntary. And then those different classes of action actually are mapped to different types of muscle tissue that are genetically determined by different classes of genes. So we kind of start to see how we can connect the different layers by connecting behavior to tissues and tissues to genes. And another important component here is that the autonomic activities in the nervous system appear early in life, and they're basically the bottom-up contributions to the developing system. And you have the top-down motor control that develops later in life, and that's where we can find these voluntary types of action. So, let's see how we can apply this to characterize and quantify the human maturation. So in the lab, it has been found, like for over a decade across different data sets, that these micro-movement spikes, they follow the gamma family of probability distributions, which is fully characterized by the shape and the scale. And because we can express the first and the second moment of this distribution very easily in terms of the shape and the scale, we have that the noise in the system is equivalent to the scale parameter of these empirical histograms. And because we also found out that there's a consistent power law that we see there, a power law between the shape and the scale, then the two parameters that fully characterize the system are reduced to only one parameter, which is very important because then in a single parameter space, you can track human maturation. So here you have the scale or the noise, and in your typical aging, in your typical development, you expect this noise in the system or this scale to the logarithmic scale to linearly drop as the system matures, something that we do not see in autism. So this is where targeted treatment should focus. Can you bring the system towards a state of motor control and decrease noise? And another application of this is by calculating this parameter space, these gamma moments, is you can kind of find self-emergent clusters in data and sort of start characterizing different disorders of the nervous system and separate them. So here you can appreciate this much better, more visually. So we see that as the system matures and the noise drops, the scale decreases, the shape increases. So the probability distributions from that gamma family that characterize the system, when the system in a state of high noise, they resemble that of the memoryless exponential because the whole thing shifts upwards. And then when you move downwards this parameter space, the probability distributions as the system approaches a state of low motor noise, they resemble sort of a shifted Gaussian. So these were the preliminaries. So now I'll start with my first published work that kind of starts building upon this precision autism model that we want to develop. So, but first we need to talk about some basic concepts. So when we talk about afferent flow in the nervous system, we're talking about the central nervous system activity that comes from the central nervous system to the periphery to execute some motion. And when we talk about afferent activity, we're talking about the peripheral sensory or proprioceptive input that comes into the system. And because those two constantly interact, that's how the models of kinesthetic reafference and loops in the system developed. And the first modeling, it started in the 1950s. There are so many models since then, for example, models for the cerebellum, but they all originate in the principle of reafference, which basically means that the nervous system takes into account the afference that arrives in the system after a specific action to kind of error correct its trajectory. The problem with these internal models for action is they almost exclusively they consider the voluntary types of motion. So you have this controller, you have some efferent copy, it goes in the forward predictor, then you compare the actual output with the anticipated output, you send the error back to the system, and the system sort of tries to adjust. But I tried a different approach here. Of course, this still holds true in the system. You have like a forward predictor, but the problem with the previous model is that these inputs that you send to the system, you assume a priori that you know the timings in the system. You know that there is some dynamical system that behaves in a good way, and we know the timings of the system. But As a matter of fact, when we measure, we don't know the timings, and we cannot assume that the nervous system knows the timings exactly before the interaction with the environment takes place. So what I did here instead, I tried to employ causality to measure this feedback loop between action and proprioception. And from these causal models, I derived an approximation or an estimate for the timings of the system by finding the time scale for which this causal influence between different body parts, this loop here, these are causal models, this loop, the causal flow is maximum. And that's how I empirically estimate the times in the nervous system. So we don't assume them, we estimate them online. And we collaborated with Columbia University and Stevens Institute of Technology, and which gave us data from 118 participants with different vestibular issues, and they had some kind of score called DHI, which is a score of business handicap. And then we also had the ASD participants, SHANK3, controls Parkinson's disease, and FMR1 carriers, which it's a genetic mutation that when you reach the middle age, suddenly you start manifesting symptoms similar to Parkinson's disease. And we saw that by employing these techniques, these causal connectivity techniques, we kind of start to see that the FMR1 carriers, that group is very different from all other groups. And the interesting thing is the participants here, of course, they had a genetic screen, so we knew they carried the gene, but they were college-aged participants. So, but our methods were able to forecast 15 years ahead of a clinical diagnosis. And also we saw that the interpeak time in this whole framework of micro-movement peaks can also be used to kind of stratify this heterogeneous cohort of the population. And here you can see this stratification process more clearly, where you have the noise-severe ratio. Here is some parameter which we defined causality over the optimal lag, and you can see we were able to stratify all these different groups. And here here you see an assessment of the whole kinesthetic feedback. So here we calculated based on our model the prevalence of feedback in the system, because you have one process affect another process, but is the other process causally affecting the first process as well? So we estimated the prevalence of feedback loops in the system and found that Parkinson's disease, FMR1 carriers, they have a lower kinesthetic reafference in the body, and also in elder controls, we saw lower connectivity in the lower body, in that part of the network, and higher in the upper body. And here you see within the normative group from the SHANK3 and the SHANK3, you also see that this timings, the system and causality, they separate the two groups. And that far we talked about times, we talked about causality, but also there is another component in this process, not just the time process, like Poisson-like spike train, but we also care about the actual values, what they can inform us of. So if you calculate the histograms for the different groups, you can see that the distributions are bimodal. You kind of start seeing how we can find subtypes in these groups. So what are the conclusions that far? We managed to employ causality to characterize this reafference in the nervous system. We stratified, and using that, we managed to stratify heterogeneous cohort of a population, which is the first step in discovering subtypes and developing the precision autism model. We forecasted a disorder of the nervous system 15 years ahead, and we defined a new notion of internal timings in the system that is not pre-assumed, but it's online calculated by the nervous system. So now we move on to further build a model and move a bit lower to the level of genomics, and that brings me to my next work, which was a collaborative work with the Salk Institute and the lab of Fred Gage and the lab of Terzinovsky. And basically, we dynamically interrogated the human transcriptome the 24,046 genes of the human tricycle in order to kind of map this to the origins of neurological and psychiatric disorders. And we did this across three hierarchical levels of inquiry. So we consider single gene expressions during the embryonic stages, pairwise codependence between genes, as well as graphical modeling of entire networks of genes. So what was our data set? We had available human embryonic stem cells that were validated in vitro, and they were matured from day 12 to day 54 when the cells matured to fully functional neurons. And the data type that we have is basically the cells, 500 cells, and the expressions of the 24,046 genes in those cells. So this is our data type, this matrix. And usually what people do when they study, they use gene expression data to discover different types of cells, different subtypes of cells, is they employ dimensionality reduction techniques, such as t-stochastic neighbor embedding, which are good for the intended purposes as method, but we believe that here, when we want to dynamically describe a system, we don't want to throw dimensions away, especially if you don't know what the roles of these individual genes. And as a matter of fact, people usually keep only 5,000 out of the 24,000 genes. That's why we decided to keep all of them. And we inverted the problem. We consider the expression of all 24,046 genes across the cells, and we tracked this their trajectory. We had available data from day 12, 19, 40, and 54. So starting from the level 1 of analysis, here we see the raw expressions, the raw data of expressions in log counts per million. That's the mRNA transcripts, these are the counts in the different cells. So for a specific gene at day 40 and day 54. And here we can normalize it inspired from the framework we already use, because we tried to develop a framework that's consistent across different layers. And this is not the time. This is not a time parameter here. I mean, it looks like a time series, but it's actually not an order. So it's just samples from gene expressions from different cells, and then we computed empirical histograms from these standardized gene expressions, and then by using the proper metric, the earth-converse distance, you can track how far a specific gene travels towards neuronal fate, or how quickly does it change its probabilistic behavior during embryonic development. And you can appreciate this in this figure. Here you see color-coded is how much it travels, how much the probability distribution changes when the system moves from day 12 to day 54. And here you can see on each individual days how the genes move in the parameter space of the first 3 moments. And we can also locate neurological, the black ones in neuropsychiatric, the green ones, disorders in this space. And you can see here that the ones that are colored very red, which are the ones that really change, their behavior really changes, the genes that behavior really changes when the system evolves, kind of cluster together, and the ones that do not travel far towards neuronal fate as well cluster together. So that becomes clear if you just see the mean and the variance here, and we see these two self-emerging lobes. These are the ones that don't travel far towards neuronal fate, and those are the ones that travel far. And that's how we discovered 2 subtypes, which we call the low expression variability and the high expression variability. The low expression variability means their state doesn't go on and off too often. And those are the ones that a lot of them are thrown away. Yeah.
But there are a lot of mixing, like green and.
Oh yeah. They distribute the neurological related genes and the neuropsychiatric related genes. They kind of mix inside.
So you find 2 trends.
Yeah, 2 trends. Yeah. But this doesn't imply yet that we can separate neurological from neuropsychiatric, because they mix in those lobes. Yeah. And precisely for that reason, we moved to the level 2 of analysis. So because we kept all the genes from a specific set of genes. We collect a specific set of genes from different sources that are associated with specific disorder. We take all the plausible n by n-1 over 2 pairs of this network of graphs, and then for each pair we do hypothesis testing. We find, are those two genes statistically independent? We can do correlation because, as we saw, these are not normally distributed, and that way we can extract dependence index that shows us how interconnected this network of genes are. So we get a global property by keeping all the genes, and the application of that, we can see it in those figures here. We see here the cumulative rate movers distance, or the fate, when the system reaches the final state of maturation. And here you see the final degree of interdependence. This is a number between 0 and 1 that shows how many pairs are statistically dependent. And here it's kind of a similar figure, but we have here with an arrow how much. It's a better visualization how much the dependency increases in this network. And we see an interesting thing here that different disorders start to kind of separate. Like we can see that ADHD schizophrenia are close in that parameter space. Makes sense because there's a lot of comorbidity between those two disorders. We see the Parkinson's disease being down here. We see bipolar and infantile schizophrenia up there. So we managed to map for the first time genes to disorders by taking global properties of.
This meeting is being recorded.
This is a and then we move on to the level of analysis, where we consider the multivariate joint behavior of all the neurological or all the neuropsychiatric genes. So we take all the neurological genes, for example, associated with neurological disorders, and we consider the joint probabilistic expression of all of them simultaneously. And then we have a vector of these probability distributions corresponding to each gene. It's variable in this vector, and then we factorize this network so that we have conditional dependencies between neighbors. But this will depend on the behavior of the entire network. So that's what we build. We factorize this graph, and this can be shown to be equivalent to a simple optimization problem, where you calculate the mutual information between the probability distributions of these genes, and then you find the maximum spanning tree of that graph. And what do we accomplish with that? We accomplish to have a hierarchical structure, some tree, where we have each gene here having some descendants and some other nodes above it. And that way we can order the genes according to their significance in that network. So if a gene is conditional dependent on a lot of other genes around, it's obvious this gene is important. But you change the probabilistic behavior of that gene, that means that it will affect the rest of the network. And we call them the key genes, the ones that have the maximal degree in the network. And here we see when we apply it in the high expression variability type of genes, we see this centralized structure where you have a few genes that have a lot of neighbors around. So these are important genes, and this structure is critical or fragile, because if anything happens, is going to happen in these genes, any mutation, that means it will propagate across the network. But when we focus on the low expression variability genes, we see that on the intermediate days they have a different architecture. Here they're more distributed. So what do we find by locating the importance of each gene in this network according to how much it influences the neighbors? Is we located those important genes, and we found out that a lot of them are aligned at the interface between the immune system and the nervous system by recapitulating the information from different sources, or they play fundamental cellular roles or roles in the different functions of neurons. So, and we don't know, maybe a lot of these genes would have been thrown away if we did dimensional reduction. So, so now let's move on to the third part, that is currently other provisional patent. So what did we do that far? What did we accomplish? We have this entire class of actions which we're able to characterize, right? With the parameter space, the Gamma parameter space, and then we have this different actions of the nervous system mapping the different tissues. But now we're able to locate, as you see here, locate genes to the different tissues. So, and as a matter of fact, we will focus on autism, right? Because the original goal is to discover subtypes in autism. Is we see all this overlap between autism and the other disorders, and these different overlapping sets there now are fully mapped to tissues, and these can be thought that the first step in discovering subtypes in autism. And you can see it here. You can see that the simple tree classification gives all these different subtypes: autism that resembles, that has comorbidity with Parkinson's, autism that has comorbidity with schizophrenia. And here you see how these different subtypes are mapped to basic functions that a lot of them are the basic building blocks of social motor agency, because now we have to talk about social agency, because we're talking about the individual in vacuum. We said the behavior, but does behavior mean if we don't take into account the complex social environment? So we need to close the loop by characterizing the basic unit of social interaction, which is the dyad. And we did this as an attempt to diagnose the Autism Diagnostic Observation Schedule. And basically, what our data here was the balance. We have both males and females of different ages. And basically, we have the child and the clinician during some session that lasts over an hour. And very taxing to the child. And we have sensors on their hands and on their torso. And we try to collect the data during this dyadic interaction and do a frame by frame analysis by having small windows of in the scale of 7 to 15 seconds. The optimal we find is 7.8 seconds, where the analysis worked the best. Window small enough so that the process doesn't change mass. But nonetheless, we're able to track this shared space between the child and the clinician through time. And how did we redefine the agency, the social motor agency within this context? So top-down motor control, we know how to characterize. This is the signal to noise ratio of this dynamical system. But what is autonomy? The bottom-up contribution, the thing that makes human beings autonomous in a social interaction. So in here we can see how we model that. So when we take the standardized data from during the dyadic social interaction, lying within that data is some timing process, like if you threshold, you get some timing process, some binary alphabet. So we see that we can approach the problem through information theory. That was the motivation, and the hypothesis here that is, if you have a system that behaves unpredictable, it cannot be, it can mask itself from the controller of the other agent, of the other system. And this is related to concepts that exist in engineering. So that's how we model autonomy. It's the entropy rate or the degree of unpredictability of an agent in a dyadic interaction. And then this balance between control and autonomy, and how much you can control yourself, how much you can escape the control from the other agent, this is what defines, this balance between the two is what defines social motor agency. So here we're able to characterize in terms of two numbers at each time window for all the sensors that are used in specific setting, characterize the social motor agency of each agent. But now we care about how each agent can affect another agent, or how much one agent leads another agent in this interaction. And we'll come back to causality, but now we don't do causal analysis between different sensor data in the body, but we try to see how one agent causally influences another agent. And for that purpose, we use the transfer entropy, which is non-parametric. We don't assume any model. And it's basically the reduction in uncertainty in the prediction of the behavior of an agent when we take into account the other agent. And here you can see we can build matrices like that, where we have the left-hand data from the child. How do they causally correlate with, for example, the torso of the clinician? And we can build this is the shared space. And some results that we got from digitizing Theorodos that kind of validate our intuition here is, first of all, the relationship between autonomy and transfer entropy. We see here that as the average autonomy of a child increases, the causal influence or the transfer entropy from the clinician of the child actually decreases. So by increasing their autonomy, there is less causal influence from the clinician to the child. And also, if we see here the histograms of these values, we see that neurotypicals have on average higher autonomy, but ASDs have on average higher autonomy variability. And here it's pretty much the same thing, except each point here is are the autonomies and the transfer entropy for a specific window in time. It's not the average for each subject. And we see that we can fit nice surfaces on this data that show how the transfer entropy relates to autonomy. And we did this both for the neurotypical and the ASD group. And here we also see other interesting results. We see in the panel A, by the way, here, red is ASD, blue is control. This rhomboid shape is males and the circle is females. And we see here that as the age of the participant, the child increases, the autonomy actually increases. So we kind of see that maybe we have a new maturation law, not for the noise of the system, but for the autonomy, for the unpredictability of the system. We see here that the clinician, there's not really some significant trend here between the autonomy of the clinician and the age of the child. We see here another important property is that there's a symmetry between whether we record our data and compute autonomy from the right hand or whether we do it from the left hand, because those are linearly correlated, but such thing we didn't observe for the control. And an important component that I kind of left for towards the end is that the problem general with autism is that we have 5 males diagnosed for only 1 female. So the question is there a higher prevalence in males, or are diagnostic tools inadequate for females due to a lack of a normative group when these things were originally developed? So motivated from a previous work in the lab that digitized the Theorodos is that males, that is known, have on average higher noise signal ratio. So that's why in our study we included both males and females, and we can see that the maturation laws control decreasing in autistics, increasing in controls. The maturation law is consistent across both males and females. And we also consider the shared spaces of all the 4 subgroups that you see here for both sexes. And what we're going to do, and another way to see that this test is flawed in that sense and cannot offer reliable diagnosis for females, is that if we focus on each subgroup, the female neurotypicals, female ASD, male neurotypicals, and male ASD, and we take the average autonomy indexes and control indexes for different tasks of the Theorodos. Here we have the different, these are classification of the different tasks of the Theorodos. The dark ones were not, the black ones, the black color were not used in this study. I mean, they were not used by the clinicians when the data was gathered. And we see here, and here with the fielding marker means that this specific task, we don't see a significant difference between the groups here when we move from this to this. And we see that for the females, the differential diagnostic power that this tool offers is not very good. We see that they kind of lie in the same area. They briefly shift. But when you see the Theorodos here on the male group, you get the sociomotor indexes in the neurotypicals, and then you see them drastically shifting when we move to ASD, which means that it's a test designed better for males. So the question here, another question is that it's too big. You can't have a child sitting for over an hour during a diagnostic test. It's very taxing to the nervous system and deprives the child of agency in that test. So by observing here, checking tasks that are characterized by low autonomy for the child, or tasks that don't differentially diagnose very well, we can sort of start removing tasks. So clinicians, in fact, to use these tools to reduce the size of the ADOS and make the test less taxing to the child. And also, beyond the fact that you can also classify, here we build classifiers. Here's with a simple support vector machine model, but this is the positive rates per subject, positive diagnosis per subject, because you can take each subject, you split into windows, then you run your model through each window, and you say, oh, what percentage of windows indicate to me that, oh, this individual is autistic, and then we can correlate those with the subjective scores of the ADOS, and we see a correlation between objective data calculated from some model and subjective data from the clinician. And so, and we validated this the classifier using the leave one out method. So with all the participants, build the model and all the participants except one that tested on the remaining participant, because we don't have much statistical power to make sure there's not a case of overfitting. And I will end this talk by kind of revisiting some issue that we briefly touched earlier. We see that there is some spike in some timing process of these micro movements. And the question is timing in the system independent of the values and the magnitude of this process? So by this is more of a theoretical work here. You can find more in the paper. We see that the gamma process and the binary process, the gamma process can be, the actual continuous micromovement process can be split into a product of a binary process and some assumed underlying continuous process in the system. And then by investigating how the entropy of the continuous component, the entropy of the binary component, they relate to the actual empirical entropy and its bounds that we calculate from the data, we found that for this specific data set, independence implies that the maximum joint entropy of the two underlying components would be equal to the entropy of the continuous component. And we found that very well a lot of samples from these recordings, they fit this condition here. So there is an indication that the timings in the system and the values of the magnitude are actually independent process. Here you see that the binary, for example, follows Poisson. Here you see the upper bound of the relation between the gamma entropy empirically calculated and the differential gamma entropy. And so what we did in the chapter, we redefined agency as the balance between control and autonomy, quantified the shared space during the dyadic interaction as intuitively as we could, and validating it. And we found some theoretical implications about the relationship between timing and magnitude in this biorhythmic activity. And then we digitized the ADOS. We offered tools for automatic diagnosis, and we managed to point to some procedure by which we can reduce the size of the ADOS and make it less taxing for the child. So finishing by saying that this is everything that I presented here is precisely what we promised, is a good start for what we promised, in the sense we try to link genotype genetics to different muscle tissues, different disorders, then characterize each disorder in some stochastic space, and then all the way up to the level of social motor ages. Thank you. Does anyone have any questions?
Yeah, I have a quick question.
Yeah.
You put together autistic kids, right?
Yeah.
But there's a whole spectrum. Did you separate?
Oh, so yeah, that's a very good question, because that's the goal we needed to do. In this study, we didn't I didn't kind of pick up of this range. I will name it this spectrum or that spectrum, but I think let me find a good figure for that.
It could be your next step.
Yeah,
if you did that, then be a peak result. Right?
Yeah,
Think
identified
what
some
different
some.
different diseases.
It's yeah. The difficulty lies with the fact that we see that the correlation with age. So we need to have more statistical power. Like we take a little bracket around 10, have 50 people from there, a little bracket, because now you already have bias introduced by age, as we saw. So it's a good idea, but it's for only 20.
Data.
Yeah, not enough data. Yeah.
Pretty big something.
Mhm.
So the big something.
Yeah.
Anyone else? Any question
From the audience, maybe,
for the Zoom?
okay.
They can't hear me. They're muted.
Julie,
Oh, I need to put this back. Yeah.
yeah,
Oh, just wondering, the committee asks questions later, right? We can wait until, yes?
yeah.
Yeah.
but
I
now
mean,
we
from
have
the general
general question.
general-
No audience, okay, fair enough, fair enough.
audience. Yeah.
Vicario has a question. David Vicario has a question.
Okay. Yeah.
Hi.
Hello. Yeah.
Yeah, excuse my my naivete or something I may have missed early in the talk in terms of the micro movement concept. Are these micro movements with the subject at rest, or are they with the subject sort of moving? And also, what can you say about the sort of magnitude of these movements in sort of absolute rather than abstract terms?
Yeah. So yeah, that's a very good question. So this is a moving task. It's any kind of task. It doesn't. The subject may sit down. They may walk around. They may do some pointing task towards some target. But basically, the intuition behind the micro movements is that when you convert to this standardized peak activity, where you have peak activity, you have a high peak, means that the system suddenly changes state. You have a sudden movement, which is the active movements, and you have quiet movements in between. So that's the intuition. It's kind of it's a way to represent in our data when there is an activity in the system. It could be speed, for example. If there's a sudden peak in speed, in this standardized form of speed, a sudden spike would mean there is a sudden initiated movement at that moment. I hope that somehow covered.
I didn't quite get the speed. Let's say there's an external event that stimulates a movement or a change in the speed of movement. How do the micro movements figure in here?
There is you will observe in the micro movements. Let's find a nice plot here. You will see a sudden peak here. You see a sudden peak. So all the values below some threshold, when you calculate this data type, are set to 0 below some. They're not the local peaks. I mean,
Yeah. So
Professor Torres developed
So-
these two.
So the quiet times are the average empirical average activity of the person
Yeah,
at that moment.
at that moment. Yeah.
For the particular context the person is. So it's a quiet times are the empirical mean of the person, the
Yeah.
empirical mean, the empirical average activity. Bursts of activity then come away from that. So it's signaling that something happened away from that. And so this is independent of resting state or just naturalistic movements.
And that's where the scaling comes, because each individual will have a different empirical mean.
Empirical
So
mean
it
for
will
each-
deviate.
For different contexts.
But you just map this to 0. You map all this range for all people. You map to 0. So yeah, it's
So you take the difference from the average.
from the
the
average.
person
So you take the
Yeah.
At the moment, so it's
Yeah.
like in the context.
In the
It could
context.
be
Yeah.
resting, but it could be sleeping, or it could be.
Because
walking around.
if you run around, then your average at that
Yeah,
local point in time would be moving. So
Yeah.
if you suddenly fall down, it doesn't mean you're at rest.
Yeah. so the point is that
Yeah.
it's not assuming a one size fits all model. Population mean assumed theoretically, it's not assuming that. It's empirically estimating it from each person. And then as a population, you see then the clusters that emerge of these gamma parameters that characterize it.
Yeah. And And it makes sense intuitively why we follow a gamma type law, because you have with low probability small movements and low probability higher, but cannot be symmetric. We don't act symmetrically around
Right. I think,
some behavior.
Melchi has a question.
One moment. Yeah. So my question has to do with, I was a little bit confused, and again, this might be that it's been a little while since I followed work from the lab, but so in the introduction, you talked about how the relative. So one thing that happens in ASD, right, in terms of your metrics, is that the relative value of the scale parameter shrinks, right?
Like you're talking about this slide right here.
Yeah. So and it just seems that what in this case, there's, I think you might have mentioned this, but there's a sense that sort of so when the scale parameter is large, that's very noisy, that's unpredictable behavior.
Yeah.
Right. And so there's a suggestion that the unpredictable behavior is a sign of agency, but it seems to be in the reverse direction of what you discussed.
I'll explain exactly. That's a very good question. Yeah, thanks for pointing that out. So because it seems like the two concepts are the same, right? In a way.
You escape from the thing and then move to the slide that you want to on the side. Otherwise, yeah.
True.
Just escape from the presentation.
I get from the presentation, yes, the presentation. Okay.
Yeah. The difference between the memory and exponential.
So
I
please
see what happened.
let me so let me go to
the PowerPoint.
I'm going to the corresponding before that. So
No, because we're not seeing it on the screen.
I'm putting it on now.
Okay,
I'm putting it on.
Uh,
So what we talk about mean by noise when we talk about the noise signal ratio, right? It's the variability over the mean. A process can have a high variability over the mean and be predictable. And a good example of that, take any cosine function that moves around a specific value. This has a high variability around the mean. You couldn't perform a stable movement if you kept bouncing periodically around a specific target. So this has a high noise, but it's very predictable. So predictability does not equal high variance in a process. Entropy rate and this they're not, unless we're talking about standard process like white noise. You cannot equate the two things because a periodic smooth movement is very predictable, but it's very noisy in the context of motor control. It's not stable. It doesn't converge to a specific target. So
okay, so this is just a characteristic. So what you discussed in the first slide, right, is really more a characteristic of how you're summarizing the data. It's not really variability. If it's predictable, it's not really variability.
no,
right? I mean, statistical variability.
It is.
It is variability in the motor in the sense of motor control.
But I'm saying periodic behavior. If it were perfectly periodic, then it's not right to say that it's variable, right? If it's periodic and it's completely deterministic.
Classified as it's not a convergent process, it's a divergent process actually. So but yeah, I understand what you mean, but this was an extreme example.
Okay.
Is the fluctuations away from the
fluctuations away from the empirical mean.
And then if they distribute exponentially, you have the memory case, which is in the human, appears to be linked to the high noise in the maturation scale.
Yeah, so
they're two separate things.
They're two separate things, and the confusion, because I understand, because I was confused at first too. The confusion comes from because we tend to think everything in terms of white noise. When you think of things in terms of white noise, then yes, unpredictability is a equivalent to the level of noise in this case. But this is not the case here. You can have processes that. But yeah, good question. But yeah, they're different concepts.
It's just that in the case of autism and the human maturation, they subsume to one parameter because of the power law. But in general, they are two separate things. You have the scale dispersion and you have the shape indicating the special case of the gamma distribution.
Yeah.
In general, they're different things, but for human maturation of the motor code and the way that we have defined the micro movements, they happen to follow a power law that reduces one parameter to the other.
Exactly.
So having the noise also predicts the level of predictability in terms of being random or being Gaussian.
Yeah.
But in general, they're two separate things.
They're two
It's just
separate
that in
things.
this particular characterization of the human biorhythms, they come together as this power law.
Yeah, one is a statistical moment. When we're talking about, and that's how we define like the variability, statistical moment. When we look at entropy rate, actually the modeling, how all this methodology comes to place is it's kind of an approximation to a discrete case. So there is a notion of an alphabet. And if you don't have a continuous dynamical system, you can create the notion of an alphabet by kind of like taking the phase space where the system moves and assigning a sort of a symbol in each of them. So they're different causes. They're related, but only in the context when you try to state for a specific probability distribution. But here it's for any stream of data, basically. So, but yeah, they're and you can see that they're not the same actually. If I have it in this slide here. So you see here, you see the control, right? This control is the inverse of the noise-signal ratio. So this is like a fluctuation away from the empirical, the variance of the fluctuation away from the empirical mean. But then you have the autonomy, which is the unpredictability of the system. And if the hypothesis that the noise is equivalent to the autonomy as unpredictability, this means that as you would go up in the scale of the control, you should expect this to drop. But there's not any consistent relationship with the two metrics here.
And that's also shown in panel C versus panel F.
Versus panel F. Here you will have this symmetry between right hand and left hand. But here it just collapses. I mean, if one symmetry is preserved here, it should preserve in an equivalent concept, but it's not preserved.
Yeah, they're almost.
They're almost orthogonal. So the data themselves show that the unpredictability as entropy rate is not the same as the noise. But that was a great question.
Yeah, very good. Any other questions?
Any?
Okay, so I guess that now
I stay here
we stay
and
with the committee,
our audience.
and then we thank the audience for joining us. And yeah, we will proceed with the evaluation. Yeah. Yeah, so